How can we figure out which animal is the smartest? We could try measuring brain size. Then the sperm whale with its 9-kilogram brain would be the smartest animal. However, it is just a huge animal. Of course, its brain would be larger, right? Maybe intelligence is determined by the relative brain size. Well, then in small mammals, that makes up about 10% of their body. In an adult human, about 2% of their body. And in the aforementioned whale, only 0.005% of their body. However, no one is really handing out Nobel Prizes to either mice or whales, are they? So what's really going on? It turns out that intelligence is not determined by the brain's relative size or its actual size. It is about how the brain is structured. Not every brain structure makes you smarter. That role is held by the part of the brain that allows for complex thought, the cortex. As animals get larger, their brains don't just scale up evenly. Instead, the cortex grows disproportionately fast, creating more surface area in a compact, limited space. This leads to what we know as cortical folding, and that is the key to our pursuit of intelligent thought. So, what are these folds?
Imagine this sheet of paper represents the brain. If I try to fit it into a really small space, I have two choices. I can either cut it up into pieces, which obviously wouldn't work for a brain, or I can scrunch it up so that more surface area fits into a smaller space. This is exactly what happens in the brain. The outer layer, called the cerebral cortex, folds into ridges, called gyri, and grooves, called sulci. These folds increase the brain's surface area and allow for more neurons and greater processing power. But why does this happen? The answer? Skull constraints. Evolution has found a clever way to maximize intelligence without making skulls unbearably large.
You might be wondering, when did cortical folding first happen? Maybe brains have always had it. Well, if you thought that, you'd be wrong. Cortical folding evolved gradually. Early vertebrates from up to 300 million years ago, such as fish and amphibians, were, and still are, lissencephalic. This means that their brain is smooth and has less complex structure. Warning, do not confuse lissencephalic brain with lissencephaly, which describes a severe human brain malformation characterized by a smooth cerebral surface and absent normal convolutions, leading to cognitive deficits and seizures. Early mammals, around 200 million years ago, started to show signs of cortical foldings, although some of them still retain their lissencephalic structures. These are rodents and hedgehogs that can be found in our world nowadays. They also have less complex connectivity and less cortical fields.
As mammals evolved, their brain sizes grew and surface expanded. But within the constraints of the skull, species began developing indentations to accommodate this expansion. This led to the development of what we call the gyrencephalic brain. This feature is mostly associated with mammals. Increased folding maximizes the brain's surface area, enabling more complex processing and advanced cognitive abilities. There are different levels of foldings. Primary foldings are the earliest and most fundamental folds that appear during brain development, and hence are generally conserved between species. For example, almost all mammals share the longitudinal fissure, which is a groove that separates the brain into a left and right half.
Secondary or tertiary foldings describe increasingly more complex folding. They arise during later stages of gestation and are thinner than primary foldings. Here you can see the different stages of cortical folding in a ferret versus a human. In some species, like the ferret, only have primary cortical foldings. In species that have secondary folding or secondary and tertiary folding, you can see that these folding occur sequentially. As the level of folding gets higher, the thinner and more defined the grooves on the surface of the brain become. These secondary and tertiary foldings are also where a lot of species-specific differences become variable. For example, Wernicke's area and the Broca's area, which arise from these folds, are unique to humans in our specialized involvement in producing and understanding language.
Whether animals have a gyrencephalic or lissencephalic brain is based on their gyrification index, which is a measure of the degree of folding and the ratio between the total outer cortical surface and the superficial exposed parts of the outer surface. Humans have the highest level of GI out of all the primates, but Hoofed animal and large aquatic animals and elephants showed that they have an even greater gyrification index than humans. Some non-human primates have GI values that overlap with humans.
While folding is often linked to brain size, differences in gyri, sulcal patterns, and cortical organization across species suggest other factors play a role. Cortical folding is influenced by cortical thickness, with thinner cortices leading to greater folding. Hoofed animals, like giraffes, bisons, or deer, have the lowest cortical thickness to GI ratio, while rodents have the highest. Animals like whales or dolphins with extremely thin cortices show intense folding but possess fewer neurons and a different cortical structure compared to primates. This suggests that cortical organization varies significantly across mammalian orders, affecting brain connectivity and function.
The cerebellar cortex is much more folded than the cerebral cortex due to its thinner folia and fewer cortical-cortical connections, which allows for higher GI values. Cortical evolution involves changes in neural connectivity, axons organization, and mechanical properties, all of which contribute to species-specific differences in brain folding. Indeed, we can see some examples of how gyrification varies across species.
Some evolutionary and functional adaptations can be found as examples. The echolocation found in bats, which have an enlarged auditory cortex but relatively simple folding, as their cognitive demand prioritize sound processing. We can also use the example of blind mole rats. Despite their lack of vision, they retain a homologous cortical organization, but their visual cortex is repurposed for auditory processing. This variation tells us while intelligence is generally higher in species with higher GI, cortical folding alone doesn't make you smarter.
At this point, you might be wondering, if animals like dolphins have more cortical folds than humans, then why aren't they holding this TED Talk? More cortical folds can mean more processing power, but it's what the brain does with that power that matters. Dolphins have incredibly folded brains, but their intelligence is specialized for echolocation and social interactions, not abstract reasoning. Elephants also have a highly folded cortex, but their intelligence focuses on memory, emotions, and spatial awareness to be able to navigate their large bodies well in their environments. As another example, cats have much smaller brains than dogs, with dogs also having a higher gyrification index than cats. However, since cats have a higher neuron density than dogs, this is associated with better cognitive processing. This leads to comparable levels of intelligence between the two species. So, intelligence isn't just about how many cortical folds exist, but how the neurons within them connect and function. While brain folding is important, it's not the only factor in intelligence. So while the five of us are sure we have plenty of cortical folds, we haven't won any Nobel Prizes either.